In this movie, we look at the mechanisms of contraction of striated muscle, the role of calcium, and the mechanisms by which it is released. In the previous movie, we discussed the structural features of actin and myosin and how ATP powers those interactions. In this movie, I show how action potentials sweep through the muscle, how the action potentials cause the release of calcium from the sarcoplasmic reticulum, and how calcium allows myosin to interact with actin. The basic idea is that normally there are blockers on the actin filament that prevent myosin from binding to actin. Those blockers are shown as X's on the lower edge of the actin filament. Myosin and actin are like lovers. They want to embrace. But there's the blocker between them. The blocker here is like a chaperone. She's there to make sure there's no hanky-panky. When an action potential is generated, it causes the release of calcium. The calcium removes the blocker, thereby allowing the lovers to embrace. And for muscle, it allows myosin to bind to actin. But what exactly is the blocker that prevents the binding of actin and myosin? How does calcium remove the blocker? How does the action potential reach the interior of the muscle fiber so that it can affect each of the myofibrils? And last of all, how does the action potential cause the release of calcium into the sarcoplasm? There are two supplementary proteins associated with actin. The first is tropomyosin. This protein lies in the groove of the actin filament and blocks the myosin binding sites. Tropomyosin is the blocker. And the second one is troponin. Troponin is a calcium binding protein that is actually bound to tropomyosin, and it's bound at various places along the tropomyosin molecule. So the tropomyosin then prevents the myosin head from binding to actin. When calcium is present, the binding of calcium changes the conformation of troponin. Since troponin is bound to tropomyosin, it pulls the tropomyosin, thereby exposing the myosin binding sites on the actin filaments. There it is. When the actin binding sites are exposed, myosin can bind to actin and cause contraction. When calcium is sequestered back into the sarcoplasmic reticulum, as we shall see, troponin changes its shape again, causing the tropomyosin to once again cover the myosin binding sites. So next, we consider how the action potential reaches the interior of the muscle fiber so that it can affect each of the myofibrils. That in turn relates to how the action potential causes the release of calcium into the sarcoplasm and thereby initiates contraction. This figure shows the features of a muscle fiber. It is packed with myofibrils. Of particular importance is the sarcoplasmic reticulum. These are membranous sacs that completely surround and envelop every myofibril. The sarcoplasmic reticulum are the sacs which contain the internal stores of calcium. The next feature is the sarcolemma, or muscle membrane. The sarcolemma contains voltage-gated sodium and potassium channels and can both generate and conduct action potentials along the entire length of the muscle fiber. The final feature is the transverse tubules, labeled T-tubules in this figure. The transverse tubules are invaginations of the muscle membrane and thus are continuous with the muscle membrane. They therefore also contain voltage-gated sodium and potassium channels. Notice that the transverse tubules extend throughout the depths of the muscle fiber and surround each myofibril at numerous places along the length of each fibril and are located next to the sarcoplasmic reticulum. The point is that an action potential that sweeps across the muscle membrane will be conducted along every transverse tubule and in this way will stimulate every myofibril. To complete the story, when action potentials invade the transverse tubules, they cause the release of calcium from the sarcoplasmic reticulum and thereby initiate contraction. The sequence of events is illustrated in this slide. The surface of the muscle membrane is shown at the top, and the invaginated transverse tubules are shown as pink tubules extending into the depth of the fiber and passing each of the myofibrils. Next to each transverse tubule is the sarcoplasmic reticulum, shown as green sacs that contain calcium. When an action potential sweeps across the surface of the muscle, the action potential then travels down each transverse tubule, causing the release of calcium from the sarcoplasmic reticulum. The calcium then allows the sarcomeres of the fibrils to contract. So the final feature we consider is how the action potential that invades the transverse tubule actually causes the release of calcium from the sarcoplasmic reticulum. This slide shows a diagrammatic illustration of a fibril. The picture that was just shown of the muscle together with the sarcoplasmic reticulum and transverse tubules is shown on the right. A more detailed version is shown by the drawing on the left. The major players are, first, the surface membrane of the muscle, the transverse tubule, calcium ions, which are shown as gray balls, the sarcoplasmic reticulum that contains a high concentration of calcium ions. The next is the calcium pump that's powered by ATP. The pump is a protein embedded in the membrane of the sarcoplasmic reticulum and operates to pump any calcium in the sarcoplasm, which is the cytoplasm of the muscle fiber, back into the sarcoplasmic reticulum. Then there is the ryanodine receptor. This is a new element in the story. Ryanodine receptors are embedded in the membrane of the sarcoplasmic reticulum, and when opened, allow calcium to flow from the sarcoplasmic reticulum into the cytoplasm of the muscle fiber, that is, into the sarcoplasm. Then there is the dihydropyridine receptor. The dihydropyridine receptor is another new element in the system. It is actually a voltage-gated calcium channel that is opened when the membrane is depolarized by the invasion of an action potential. It is called a dihydropyridine receptor because dihydropyridine is an antagonist for the channel. That is, it prevents the channel from opening. The gate of the receptor is indicated by the plus sign on the ball in the middle of the channel. The unique feature here is that the gate is actually physically coupled to the ryanodine receptor. It is shown here as a plunger that plugs the channel of the ryanodine receptor. Thus, when the gate of the dihydropyridine receptor is opened by depolarization, it physically removes the plug from the ryanodine receptor, thereby allowing calcium to flow from the sarcoplasmic reticulum into the sarcoplasm. So here is the entire sequence in slow motion. We start with the action potential shown at the top left. The action potential will sweep across the surface membrane of the muscle, and into the transverse tubule. It will depolarize the membrane, and the positivity on the inside of the membrane will affect the gate on the dihydropyridine receptor and thereby remove the plug from the ryanodine receptor. The ryanodine channel is now open, and as a consequence of that, calcium flows from the sarcoplasmic reticulum into the sarcoplasm. The calcium, of course, will then bind to the troponin. The troponin will pull the tropomyosin away, exposing the binding sites, and the sarcomere will shorten. A moment later, the muscle will repolarize. As a consequence of that, the ryanodine receptor will be plugged again. The calcium pump will go into operation, pumping calcium back into the sarcoplasmic reticulum. The tropomyosin will once again block the binding sites, and the sarcomere will relax. That is the sequence of muscular contraction.